What's going on, Kat?
Hi, how are you, Kirko? Yeah, I was just adding the topics. I always check if there's maybe new like names for stuff that is more accurate, like germs or microbes, but there's not.
Legit.
So I'll do this. Hi, Paul, how are you? It worked. So to unmute is the button all the way on the bottom right hand. There's a little microphone symbol. If you press on that, you unmute, and then we can hear you. If you can hear us. I hope you can hear us.
Okay, can you hear me?
Ah, there. Yeah, perfect.
I was trying to do it on my desktop, but that was clearly a mistake. So thanks for helping me out.
Oh yeah, the desktop version. I really don't like it, and since it's not the screen share, I prefer the phone because I can walk around. I'm not good at sitting a long time. So,
There you go.
yeah, I'll so as far as I know, I'll post a paper
Yeah.
for people to like check it out. So I shared well, for now I might share, since we are not starting, we have a few minutes. I'll share your lab website because I think it's very interesting for people that want to know more and read about different types of research you do.
Great.
So it's up on the link, and we have a bunch of minutes. We have like around 6 minutes. So in the meantime, I'll share on Twitter and so on that we're about to start. So you have a few minutes to relax. So.
Okay, and then how does it work? Is it more of a Q&A, or you want me to talk for a certain amount of time?
Yeah, so on top of the hour, I'll introduce you a little bit. Like I'll give a short summary of like, where you went to school and things, so people know a little bit about you.
Okay.
Because this is a public room, so anyone that's currently on Clubhouse or sees it on Twitter can just join.
Right.
So and then we usually ask like a few interview questions. There's more like general public type of questions, like how did you choose science as a career, like type of question, if that's okay
Yeah.
with you. Like, was it maybe a teacher, a book, or you always had the dream of, like, whatever the answer is interesting, I think. And then how you kind of chose this type of research area, and then also if there's kind of a background story about this specific, like about this project that we are talking today. But if you would like, you can also talk more in the beginning, more general about
Yeah.
the research you do in general, and then kind of go into a little bit of the paper. Yeah, and then usually after those type of more general questions, people give like an overview of their research and then also a little bit about the paper, and then people ask questions. That's how it usually
Okay.
goes.
So, so you'll prompt me a little bit, and then I'll talk. Is that the basic format?
Yeah, exactly. But if you would like then to have a part where you more talk, like a general audience, and go into the paper a little bit in between, I think, yeah, if you would like to do that for people that don't know your research at all, that's
Yeah.
also
Yeah. First, I'll do all that. Right. The general part. Yeah. Great.
cool. Yeah, thank you for doing this and for joining Clubhouse and so on.
Yeah.
Really
Thanks for the
appreciate it.
invitation.
I'll hope you'll enjoy it.
I'm sure.
Don't get addicted to this. Like, it was easy during
COVID.
during COVID to get when you're stuck at
Yeah,
home. Anyway, that's how kind of this app I think got big for a while,
I bet.
and then yeah. So I think now I don't know how is Kirko. Do you and meet Kirko? By the way, he is
Hi.
here, one of our members, and Victoria is here. So hi, Victoria. She will ask the interview. She is usually the interview
Hello.
person.
Thank
Hello.
you. Good morning, Katarina, Kirko, Paul. Nice to see all of you.
Good to meet you.
Yeah, I was just telling Paul about not too much, but Clubhouse in general. Like when there was COVID times, it could get really addictive. But
Hours, the
Yeah,
entire day,
I bet.
running from one room to the next. Really, it felt like that. It felt like it felt either like being in college or at summer camp sometimes, where you're just running from group to group, or a really great conference.
That's awesome.
And it still is. Now maybe we've learned some self-control though, right?
There you go.
Maybe.
yeah, the unique thing I think is that our members that come here all the time are really around the world. Like I don't think I would have met like researchers and so
Oh yeah, I'm
from so
sure.
many different countries and had conversations with them, and then also from different fields, right? Usually when we go to conferences is about we talk more or less about the same we are doing.
Right.
So, yeah. Hi everyone. We will start in 1 minute already. It passed fast. We had stuff to cover. Please feel free to share the room with. I have. Hi Denise. How are you? Meet Paul.
Hello.
Pleasure to meet you, Paul. Hi, Katerina. Hi, everyone. Really excited for the talk today.
Hi, Denise. Good to see you.
Okay, sorry everyone. I was just sharing everywhere that we are starting, and I guess people will still come in, but let's start with introduction and then we'll go from there. So, welcome everyone to Science Society, and of course, a special welcome to you, Paul. And as I said before we start, let me give a short introduction so people get to know you a little bit or the research you do. Um, so, Professor Paul Hergenrother was born in Akron, Ohio, and he attended the University of Notre Dame, where he did his bachelor in chemistry. And then he did he did graduate research at the University of Texas at Austin in Professor Stephen Martin's lab, where he worked on discovering the catalytic mechanism of phospholipase C, and he completed a total synthesis of erythromycin B, and he received his PhD in chemistry. Um, doing that, and then he moved on where as a postdoctoral fellow to Harvard University um, at the American Cancer Society, where he worked in the lab of Professor Stuart Schreiber in the Department of Chemistry and Chemical Biology. There at Harvard, he was involved in the development of small molecule microarrays as a platform for high throughput compound screening, which is really useful, I can say. And then he established his own lab in the Department of Chemistry at the University of Illinois at Urbana-Champaign in 2001. And then he was promoted to associate professor with tenure in 2006 to full professor in 2010. And then he was named 2013 as the Kenneth L. Reinhardt Endowed Chair in Natural Products Chemistry. And he's also the co-founder and chief scientific officer of Vanquish Oncology. And he developed anti-cancer compound there that was discovered in his lab, the PSA1, and it actually completed like the first phase 1b2 clinical trials started in February 2021 in trial patients, and he did many more really amazing research and developments that will help us humans, which he will for sure talk about here during his talk. So, and he won many different awards. So it's such an honor having you here. I could go on and on talking about your achievements. But I want to welcome you and give Victoria the opportunity to ask you a few interview questions. Thank you.
Sure, great. Thanks for having me.
Victoria? Oh,
Hi, sorry. Yeah, I was navigating back to my screen because I'm trying to bring Dr. Shaw up, but I don't have that option for some reason on my
oh, thank you.
when I
I'm
click.
doing it. Don't.
Okay.
Thank you, Dr.
Yeah.
Scheidler.
All right. Oh, good. All right. Now the party can start. Dr. Shaw has arrived. Okay. So, yes, thank you. Thank you, Katarina. And welcome, Paul. Science Society is so happy to have you here. And yeah, we really look forward to hearing about your research. And in addition to just hearing maybe a bit about you. And so, somewhere along our lives, we develop affinities and interests. And so, perhaps you can tell us about when you first noticed that you had a particular interest in science, and it doesn't need to be as an adult. It could be something, in early play, or something that somebody shared with you that that just kind of got you started in feeling interested in science.
Yeah, thank you. It's a good question. I mean, I think really for me, it wasn't probably until college. I mean, I'd always kind of been interested in science and math to the extent that any teenagers are interested in such things. But I started out college as an engineer, and I really wasn't very good at it. I didn't like the classes, and I wasn't good at them either. And as part of those classes, though, I took organic chemistry, which most people don't like, and I did like it, and so I switched my major to chemistry, and that's kind of been a big decision for me.
That's amazing. So it's kind of serendipitous, and that worked out for you that way. And
Yeah, yeah. I think I tell my own students and my kids, it's good to take classes that you don't know anything about, right? I mean, I never heard of, really didn't know anything about organic chemistry except for the reputation. But I think once I took it and I realized as a chemist, you can make things no one else has ever made, right? You can make totally new compositions of matter and hopefully can aim those compounds towards really important biomedical problems. That really fascinated me.
Yeah, well, grateful that you had that experience, and it is interesting. I mean, and it is, I think, I don't know, maybe if the method of teaching were different or people had a longer time to incorporate all the information, then OChem would have a wider reach, because it is fascinating, and and it's all volatile, so that's exciting just right there. And what you said about synthesizing new compounds, what power.
Yeah, exactly.
So yeah, it's great. Yeah, and that's, yeah, and you remembered that. So maybe you can bring us from that point up to where we're at in your current research, some events along the way.
Yeah, I mean, once I made that switch of my major, and then, I was fortunate at the University of Notre Dame to be able to take graduate classes when I was a senior undergrad. And that was also very transformative and gave me a peek on, how much more there is to learn. Sometimes we think as students that undergrad degree is really the pinnacle, but then you realize that there's a whole bunch of stuff that they haven't even told you about that's out there. And so the ability to take those classes was great, and turned me on kind of to the idea of going forward and getting a PhD. And from there, it was trying to acquire these skills. I mean, to a certain extent, science is a craft, right, and a trade, and you have to learn. There's physical manipulations and techniques that one has to become proficient at, and that's just, graduate school is an amazing time for that, to learn from practitioners, and kind of have the freedom over the course of 5 years to explore a bunch of different techniques, but then really become an expert. And then my time as a postdoc also equally transformative, learning how to then aim those skills at really important problems. And so when I set up my own lab here about 20 years ago at the University of Illinois, I wanted to bring the power of making new compounds that organic chemists can have and do have to what I think are the really some of the most important biomedical problems. So drug-resistant bacteria, which we'll talk about today, but also terminal cancers. Those are the two areas that we've spent the most time on.
Right. Yeah. Thank you. Well, at this point, then, please begin your talk, and we're here to moderate and bring up guests who may have questions, and also share questions that are placed in the room chat. And so you can either have a Q&A following your talk or have questions drive your talk, either way. And so I pass the mic to you, and thank you so much.
Okay,
And yes, science is a craft and an art, and couldn't agree more. Thank you.
thank you. And I think, I'll plan to talk for 15 or 20 minutes, but if there's things along the way, questions that you see that people have, feel free to break in, no problem. And then, of course, we can talk more about everything at the end. So I was invited to this based on a recent article of ours on a new compound that we discovered that we call Fabimycin, and it's a new antibiotic. And so I'll just start a little bit with the kind of the motivation for this, and then our discoveries along the way that then led to this event. So our real motivation here is on what are called gram-negative bacterial infections. And these are different from other infections, say staph infections, based on the type of bacteria. So there's gram-positive bacteria or gram-negative bacteria, and they're named so because they take up a certain type of stain. But kind of operationally, the difference is that gram-negative bacteria have an additional outer membrane on them, and this makes them very, very impermeable to antibiotics. And as a result, basically, our antibiotics that are really good for gram-positive infections and are widely used typically have no activity against gram-negative bacteria. And in fact, it's been 50 years since a new class of antibiotics was FDA approved for treating gram-negative pathogens. And I think everybody recognizes that bacteria become resistant very, very quickly. And so 50 years is actually quite a long time. And the net effect of this is that a lot of the antibiotics that we use to treat gram-negative infections don't work, and especially in certain places and certain infections where we've gone back to antibiotics of last resort that are actually highly toxic. And so there's a real recognition of this problem that we need more gram-negative antibiotics, but it had just been really hard to discover such compounds. And there's a lot of blood in the water from the pharmaceutical industry, and they've been great in publishing their negative results, which doesn't always happen. And so GlaxoSmithKline and AstraZeneca especially have published how they've screened millions of compounds, say 3 million compounds, looking for antibiotics against gram-negative pathogens, and have come up with zero compounds, so 0 for 3 million. So really, and these are by experts in the field and big teams of people that are really good at this. So puts into sharp focus how challenging this problem is. So a number of years ago, about probably 10 years ago now, we started a program here at the University of Illinois in my lab trying to just understand what types of compounds might get into gram-negative bacteria, not even talking about antibiotics, just like asking the question, hey, can organic compounds at all even get through this outer membrane and accumulate in these pathogens, recognizing that this is the first step in this process of killing the bacteria. And so I give a ton of credit to my student at the time, Michelle Richter, who took on this project, very, very challenging project. And I think, we took what we called an unbiased approach, where we just assessed compounds and just asked the question, do they get in or not? And I think the first 100 or so she tested, maybe one of them got in, and I was really excited about that one, but she was like, well, it's going to be tough to make heads or tails of this data if we don't have many compounds that get in. So she persisted and persevered through this, and we ended up with a good collection of compounds that both did get into these gram-negative bacteria and also did not. And so we used that data set and we used machine learning and computational analysis to really tell us the chemical features that compounds need to possess to get through this outer membrane of gram-negative bacteria and accumulate in the cell. And so that was our first discovery in 2017. We call these the entry rules, and they give medicinal chemists and microbiologists kind of guidelines for how to build an antibiotic that will actually get into gram-negative bacteria. And so we have been working since that time to apply those entry rules, apply those guidelines to develop new antibiotics. And so this paper that came out this summer is our use of those entry rules to develop a novel antibiotic called Fabimycin. So in this case, we started with a compound that inhibits an enzyme in fatty acid biosynthesis. So this is a really a new type of target. Most antibiotics out there are ones that, like penicillin, operate on the cell wall, or there's macrolides that prevent protein synthesis, and there's fluoroquinolones that disrupt DNA replication. But there's really not two, those are the kind of the dominant mechanisms. And so this target, inhibition of fatty acid biosynthesis, had been reported by others as a really promising target. There's a drug in phase 2 clinical trials, an antibiotic that hits this target, very effective against staph, but it had no activity against gram-negative bacteria. And so we thought this was a perfect opportunity to apply the entry rules, where we could take that drug that has kind of this staph-only or gram-positive only activity and modify it using the entry rules and create new versions that would now still inhibit this fatty acid biosynthesis, but now also get in to gram-negative bacteria and kill those cells. And so this has been just a really remarkable collaboration with colleagues of ours at the Broad Institute in Boston, and also Walter Reed, and also the NIAID, and bringing the powers of all those places to bear on this really important problem. And so in our manuscript, which I think has been put on the Clubhouse site, we talk about how we constructed a handful of compounds, 10 or 11 compounds, that follow the entry rules that we thought might be candidates as a new antibiotic. And through the testing of those compounds, we discovered this novel drug, Fabimycin. This is a compound that, it has, we show to have activity against gram-negative bacteria, including hundreds of clinical isolates that Walter Reed was able to assess for us. And I think one of the really interesting things about this compound and this target is the selectivity that this drug has for pathogenic bacteria versus what would be called kind of good or commensal bacteria. There's an increasing recognition that our current antibiotic arsenal is really a kind of a carpet bombing of our gut microbiome. So we have our gut is teeming with different types of bacteria. We're kind of in this symbiotic relationship with, and in general, those bacteria are great for us. They help us in many, many ways, from everything from digesting food to fighting off infection, and the immune system, and everything in between. And so it's now pretty well accepted that when you take most all antibiotics, especially broad spectrum antibiotics, you're wiping out a huge percentage of this gut microbiome. And what happens then is bacteria that are typically kept at bay can then flourish in the absence of these commensal or good bacteria. And this can lead to all sorts of problems, and including infections with Clostridium difficile, C. diff, which can be lethal and very challenging to treat. And this is why fecal transplants have become kind of an accepted means of treatment, where you're literally trying to transplant in that microbiota. And so the vision with these kind of next generation of antibiotics is that wouldn't it be great if we had drugs that just killed the pathogenic bacteria, and specifically, and of course that's a big challenge because bacteria have common ancestors, and a lot of their machinery is very similar between pathogenic and these so-called commensal or good gut bacteria. But this particular fatty acid biosynthesis target presents an interesting opportunity, because in certain pathogenic bacteria like E. coli and Klebsiella pneumoniae and Acetobacter baumannii, the enzyme that is critical to this fatty acid biosynthesis is actually quite different than the version that is in the commensal organisms. And so when we assessed this compound against pathogenic bacteria, Fabimycin is quite potent against these bacteria, but when we assess it against commensal bacteria, it actually has little or no effect. And so this is super exciting, and this is something that we're continuing to investigate. Really, the ultimate vision would be a gut microbiome sparing antibiotic, and kind of a targeted therapy almost for that kills the pathogenic bacteria, but not the commensal good bacteria. So that was a key part of the logic and of this discovery. We then kind of moving forward in the characterization of this compound, Fabimycin, is an assessment of the ability of bacteria to become resistant to it. And this is part of any sort of drug discovery platform and process for antibiotic development. We know that bacteria have the numbers on us, right? There's billions of them in a flask, and they can have mutations that enable them to resist most all antibiotics. And in fact, in most cases, it's inevitable. And so one needs to be smart about how one doses antibiotics, and where you kill all the bacteria and don't leave certain ones around that will be resistant. And so one of the things that we do is assess the propensity or the ability for bacteria to become resistant to antibiotics. And certain antibiotics is very easy for them to become resistant to, and this can be, as you can imagine, a big problem in the clinic. And those compounds typically either don't become drugs or are used in combination with other drugs. And there's other ones, and Fabimycin is in this category, where it's relatively hard for them to become resistant. And this is due to the precise means by which the drug Fabimycin interacts with this target in the fatty acid biosynthesis pathway. So that was another, I think, really important discovery, was that the frequency of resistance is really quite low for this drug. So with these kind of, these are in vitro experiments just with bacteria in culture. And then we move to assessment of the ability of the compound to actually rescue animals from infection. And so these are all kind of as a prelude, hopefully, to what ultimately will be a human clinical trial. There's really no other good surrogates. The way drugs are developed and advanced is one typically looks at analogous studies in laboratory mice as a key workhorse, and uses that as a triage or a way to advance the proper compounds. So compounds that have efficacy in those animal experiments are moved forward. With antibiotics, it's actually a pretty good situation in that mice can become infected with the same exact bacteria that we do, so that we become infected with. So we can run those experiments as simulating a human infection, either pneumonia or a sepsis or a UTI, and then treat with our novel compound. So in this case, we treated mice with Fabimycin and showed reduced bacterial burden in the lung, and then also did a UTI model, a urinary tract infection, which is a quite challenging model, and also one where humans get these infections pretty frequently, and they're typically gram-negative infections, and so it's a great proving ground for new antibiotics. And so again, we infect the mice and then treat with our compound and look for reduction in bacterial burden relative to controls and relative to kind of standard of care drugs. And so in all those experiments, Fabimycin looks quite promising. So the development challenge for antibiotics is significant, just because all sorts of really interesting reasons. There's scientific ones in that there are antibiotics that physicians are comfortable with. When someone is resistant to an infection, they need a treatment, ASAP, basically. Unlike oncology, where you might have a kind of a oddball cancer, and everyone can travel to a certain site, to the Mayo Clinic or Moffitt Cancer Center, to be treated because it's not acute, meaning in the hours or days time frame. With infections, it is actually acute, and so it's just harder to run those clinical trials because patients are kind of all over the country. Then there's also the economics for how they're paid for, and we've all become very used to paying very little or nothing for antibiotics. So there's always challenges, but we're hopeful that we can continue to move them forward. And I think now companies and the government and foundations are all aligned in their vision and their goals. And so I think there's reason to hope that there'll be more antibiotics coming through the pipeline in the next several years. So I'll stop there and take any questions.
Yeah, thank you so much for sharing this with us. And yeah, if anyone has questions, please flash your microphone and let me also, and please also share in the room chat if you don't want to speak. Yeah, welcome, Lady Rocket, and yeah, Dr. Shah, Kiku, Denise, Roy, Joyce. Yeah, go ahead. Hi, Lady Rocket, how are you?
Yes, I am doing very well. Thank you for, as always, introducing phenomenal speakers who do such a meaningful research and progress for our humanity. My question to Dr. Paul, and first of all, thank you and congratulations on your accomplishments and what it can mean to us humans on Earth. But my question has to do also with space application. Being a space entrepreneur and innovator, I am curious and being aware of NASA and private interest in finding a way to contain bacterial
I can't hear you, Lady Rocket.
containment
I
of
don't know if that's
bacterial infections.
Lady
Yes.
Rocket. Excuse me. We didn't hear you for about the last 30 seconds.
Okay.
Maybe you could rewind a bit. And welcome. Glad to see you.
Yes.
It's been
Thank
a
you. It's
while.
always pleasure to see you and be part of your room. My question, I am a space investor and entrepreneur, so allow me to diverse a little bit toward the application of your significant research to specificity of space applications, where one of the critical path items includes containment of diverse bacterial infections possibility. Let it be we are getting ready to return to the moon for the extended stay as a part of Artemis project. If you can comment on that. And the second question is, how do you look, where would you like to seek um, commercial funding for your research? Because my platform is organizing funding for the research relevant to space that traditionally is not easily supported, let's say, by venture capital or traditional investment sources. So, number one, space application, and two, sources of funding for the follow up that you would like to see. Thank
Great.
you for letting me share.
Yeah, thank you. Thanks. Those are really interesting points and questions. I mean, the space one, wow, that's super, super thought provoking and interesting. I mean, I assume and I think that there are a battery of pharmaceuticals that one needs to take in space. I mean, I think we all know that, you know, people that go to space have their appendix out already, right? And so they're trying to hedge against certain things that are relatively common here, but obviously would be life-threatening in space. I think antibiotic or bacterial infections would be in that category, right? With things that we know how to treat, but if you're in space and don't have access to antibiotics, it becomes life-threatening. So yeah, I think there's definitely drugs one needs, and antibiotics would be at the top of that list. Um, uh, I think a drug that is gut microbiome sparing would be even better, right? Because as I mentioned, the challenge with some antibiotics in our arsenal is that in some people, now it's typically people that are in hospitals, but in some people, this wiping out of the commensal bacteria can lead to secondary infections, fungal or bacterial infections. So I think it's a great question and problem, and someone needs to make a decision about which drugs, antibiotics go to space. I think the second part, yeah, the funding, I kind of hinted at that towards the end. You know, it is a challenge to advance antibiotics, but fortunately, there are public-private partnerships and enterprises that have been assembled recognizing this problem. Companies have put money in, governments have put money in, and trying then to move them forward. I mean, the typical path would be from a university discovery like this one, the typical path would be a license to a pharmaceutical or biotechnology company, and then they're now it's in the hands of professionals, and they would move it forward. I think there's other possibilities though, especially for antibiotics, where potentially even with the government getting it into phase one, and then really seeing getting a basic look at the kind of the pharmacokinetics and efficacy, and then maybe going to a commercial entity after that. So I think also, there's all sorts of possibilities for Fabimycin. The pathway has not been decided.
I want to just add one more that would potentially allow you to retain ownership because I suspect maybe you have consideration to become an innovator, researcher, entrepreneur. Our platform, Copernic Space, is providing funding through utility NFTs to space endeavors, including as far reaching as financing first private mission to the moon. And I am mentioning it because there is a revolution in sources of funding introduced by tokens, NFTs, blockchain that liberate or extends choices for synergistic with your mission sources of private and public funding. So that's another, I would say, opportunity for research as significant as yours. And last question, is Eli Lilly potentially one of the better choices for industry partner, for pharmaceutical company as a partner?
I can't, I don't really, I wouldn't be able to say. I mean, different big pharma where Eli Lilly would be considered one have been kind of in and out of the antibiotic discovery process over the last 40 or 50 years, and it kind of depends on different strategic directions within the company, and I don't know where they're at with that right now.
Okay, thank you so much. I appreciate this room, and thank you,
Thank you.
Katarina.
Thank you.
Thank you, Dr. Paul.
Yeah, thank you. Kiko, did you have a question?
Yeah, I was kind of curious. So, like, as you go through this process of discovering, creating the mycin drug that you guys created for these gram-negative bacteria, was it also in the thought process on developing something that could be made like effectively and cost efficient? Because one thing that I think is kind of the cruxus of modern medicine is that sometimes you have a much needed um, medicine, antibiotic, what have you. The price tends to be so far out of reach of common folks that a lot of common folks end up losing, you know what I'm saying, faith in the medical industry.
Yeah, yeah. I hear you, and I think it's you're kind of breaking up, but I got the gist of it. I got the gist of it, which is basically, can one manufacture this compound at a cost, a scale, and with cost of goods that would be make it into a reasonably priced drug, and also asking about how that is maybe not the case with some other drugs. The answer is yes, absolutely, and I think that when you look into the cost of goods for drugs for what I'd call small molecule drugs, so these are organic compounds, which are most all antibiotics and many, many other drugs, is the amazing ability of chemists, medicinal chemists, process chemists to come up with really, really efficient synthetic routes to these final products. And actually, the cost of goods is a fraction of the total cost. So that would be the situation for this compound as well. Now that changes a little bit if you go to some biological therapies that are used in other indications, cell-based therapies. Some of those the therapy itself can actually be very, very expensive. But for organic compounds, making those on scale can be done very, very efficiently. And so then what they're priced at, of course, is different and based on other factors. But the actual cost of goods for the drug will be nominal.
Super cool. Thank you.
You're welcome.
Yeah, thank you for the question. Joyce, Dr. Shah, Denise, do you have questions? I now joined the stage.
Yeah, thank you so much, Paul. That was wonderful research that you just shared with us. My question from you is about the using of the nanoparticles, and we have a recombination. And I was just wondering, as an next step, because we have a use of that, or we might have a use of that in immunotherapy in the future. And I was just wondering if you have further information around that.
Yeah, so the question about nanoparticles, and those are used to formulate drugs in some cases. One of the challenges, one of the interesting things is that most of our drugs, as I mentioned, are organic compounds, meaning they're carbon-based molecules, and they tend not to be soluble in water. And so there's a little bit of a tension there because ultimately they have to get into circulation. So one of the solutions that has been come up with is to use something like nanoparticles. In some cases, nanoparticles can help solubilize. In some cases, they can help target the compound, say, in certain cancer applications. I would say, the nice thing about, so our entry rules, I didn't get into the specifics, but one of the key parts of that is the presence of what we call an ionizable nitrogen or a primary amine. An amine is a group that is positively charged at kind of standard pHs, and that greatly facilitates solubility in water or aqueous solutions. So this compound, to answer the question directly, this compound is such that one wouldn't need a nanoparticle formulation. Thank you.
And is there any side effect that you just came up with?
I think, right now, all the toxicity profile has looked very promising. The work that we've done in mice and some other work that we've done in rats and then other kind of cell culture based assessments. But one doesn't really know until you do the kind of the toxicity studies that one needs to go into phase 1. So I think there's reasons for optimism, but it hasn't been explored to all the way yet.
And what's the age that you clinically you can use it? For example, in what age there's supposed to be harm?
Age restriction for the drug. I mean, certainly that hasn't been decided yet, but I think most of these start out as adult indication and then kind of get assessed in younger cohorts after that.
I see. Thank you so much.
Thank you.
Hi, Paul. I was curious about so you just mentioned the animal studies. I was curious about the timeline to human trials and then perhaps from human trial, basically the bench to clinical setting.
Yeah, that's a great question. I mean, in general, it's all related to how many resources can be deployed. I mean, as I said, the most straightforward path for any new drug is from a place like ours, from an academic institution, is a license to a company that could be a startup, or even better, probably for the technology, would be a license to an existing company, right, that has the resources. So it will cost maybe a couple $1 million to do the experiments that one needs to do to get into the human clinical trial. So, but those can go relatively fast. So if, in a situation where one has those resources, you can probably get into a clinical trial on kind of a couple years time frame. And then again, the clinical trials, if one is really pushing on them, with antibiotics, everything is pretty well understood what one is looking for, and there's nice blueprints and pathways, so patients can have access to the drug through the trials. And then, in some indications, things can be granted, drugs can be granted accelerated approval or breakthrough designation if you have a drug for a indication for which there is no good drug. These are typically in oncology, but it's possible in other diseases as well. So, what might be a 5-year process could be kind of shrunk to half of that.
Thank you. Who would like to go next?
Thank you.
Hi.
Go ahead.
Yeah, this is Joyce. Thanks for your presentation, and it's great, exciting work. I was wondering, there's increasing evidence that microbes can, it's well known that they can transform into L forms or cell wall deficient forms and become latent and then potentially come back later and cause problems. I was wondering if you think this antibiotic, some antibiotics actually
enhance the formation of L forms or cell wall deficient microbes. But do you know about that in this case? Thank
Yeah,
you.
that's a really interesting point and questions. Uh, no, we haven't studied that. Okay, so that would be my short answer to your question. I mean, I think, you know, what we have looked at is the bactericidal nature of the compound, and also the ability to give high doses that are above the minimum inhibitory concentration. So when one can dose at higher than you need, say 5, 10, 100x higher than is needed, then you have great confidence that you're killing the bacteria quantitatively, so you're killing all of them. Now, the persister phenomenon that you're mentioning I think, is really fascinating. It's a kind of an area of research that, man, there's just so much more to learn, I think. So it would be something to look for in the future, definitely.
Yes, thank you very much.
Yeah. Thank you for answering those questions. I have a question. So you target, like you mentioned that, or also in the paper, that amine is like essential part that you kind of, that is targeted. Are those aren't those like pretty like broadly used, and like, is it specific to bacteria, or to or could it also target other cell types, including fungi? Because we also have a huge fungi problem, I think.
Yeah,
Uh, yeah.
yeah, yeah. It's an interesting question. So so when we developed the entry rules, we were looking for chemical traits that enable drugs, compounds to get into gram-negative bacteria. That's kind of very specifically what we were looking for, and we came up with three things in the N, the T, and the R in entry: the nitrogen, the amine, and then the kind of what we call three-dimensionality. So the ability of a compound to be flatter, relatively rather than spherical, and then the rigidity is the R, rotatable bonds. And so a compound that has all of those three traits is really predisposed to get into gram-negative bacteria. I think the entry problem is not so acute for gram-positive bacteria or even for fungi. The drug development challenges there are significant, as you're pointing out, but for different reasons. So the so it's not to say that the compound would only go into gram-negative bacteria, but the nature of the target, this fatty acid biosynthesis target, is such that, in a best case scenario, it will only restrict the growth or kill gram-negative bacteria, and if it does go into other cells, it would be innocuous.
Yeah. And are you like, when you came up with this compound, it was you know, purely human based logic, I guess. So how do you feel about the approaches of using machine learning slash kind of AI to find different compounds? Do you
Yeah.
believe in them, or do you think it's really hard to tell what the correlations they make, if they are really trustworthy and reliable?
Sure. Yeah, I mean, I think machine learning and artificial intelligence, it's another tool, and it's a great one, and it's a powerful one. And another tool that we have in our arsenal, as I mentioned, it was critical, that tool specifically was critical for us in our development of the entry rules. And so that's really, in drug discovery, that's a powerful application of machine learning and artificial intelligence is giving it big data sets that we as humans really have a hard time making heads or tails of, where we have hundreds of compounds, each with hundreds of chemical features, and trying to understand what features are common to ones that accumulate and ones that don't, right? And especially when you get into a combination, it needs to be 3 features or something. So I think that is a great role for artificial intelligence and machine learning. But that said, the roles are expanding all the time. I mean, there are now algorithms that can start to predict chemical routes or suggest chemical routes to compounds, which in some cases are competitive with even the best chemists out there. There's computational methods to actually discover enzyme inhibitors, so drugs that bind to certain important targets. So all these things are tools, and they work to a greater or lesser extent depending on the specific application. That there's a that's the human element though has never been more important, right? I mean, it's like every time the tools get better, one needs to know how to use them. So I think that being up on the latest tools is critically important, and being able to use them as a scientist is critical to making discoveries that can be made rapidly and that are also impactful.
Yeah, thank you. And was this also, like, do you use the approach also for your cancer-related drug research development?
Yeah. Again, I think it's conceptually some of the things are similar. Trying to find compounds that, in that case, have selectivity for cancer cells over mammalian cells. In some ways, the cancer challenge is even more significant because a cancer cell is still is 99.99% similar to the human, the non-cancerous cell. So that's it's very challenging, and I think in that case, computational analysis has been really instrumental in discerning what the differences are in cancer cells relative to normal cells, and what as chemists one might exploit. So those tools are critical, and anytime a new tool comes online, it's a really great day for. It's like CRISPR or some of the newer structural biology techniques that just can really transform and enable scientists to do their job faster and better.
I know. Yeah, thank you so much. It's exciting that it's apparently giving us so much new possibilities to fight off cancer, germs, and so on. So yeah, thank you for doing this research. And Einar, did you have a question? You came on stage.
Yeah, hi, Katharina. Hi, Paul.
Thanks.
I have a question. This is not my field of work, so be brutal. But when it comes to this research, Fabimycin, if I understand this correctly, does it have a tendency to actually relate to nitrogen-related molecular bondings in a different way that we've been seeing earlier? When it comes to, I don't know if this is the correct way to see it, but that we can either recycle or we can destroy or we can reuse what we have as a chemical compound or normal matter, if I can use that word, in our bodies. Does it work differently when it comes to specific fatty acids containing some form of nitrogen bonding? Yeah, thank you.
Yeah, I mean, the fatty acid biosynthesis pathway in bacteria is really fascinating, and there, there was a thought, which maybe is what you're getting at, that if one inhibited fatty acid biosynthesis, the bacteria could actually get around that by recycling components that they already had, cell wall or products of those cell wall biosyntheses. So until the first FABI inhibitors were developed, that was a question. And then even going into gram-negative bacteria, when we discovered the first version of these a few years ago, was definitely a question. But now, with these probes and others, that question has been definitively answered that, yeah, if you inhibit fatty acid biosynthesis in these bacteria, they will die, right? That they cannot bypass that process. By uptake of say, intermediates or by shuttling, degrading things that they have. It's an essential process. So yeah, I think it's an important point to bring up. Thank you.
Thank you.
Hi, Watch Dominion. Welcome to the stage. Did you have a question?
Oh, yes. Hi, Katerina. Thank you so much for giving me the chance. I have a question regarding the amount of antibiotics being used in factory farms. And I know there is several antibiotic resistant strain of staph bacteria, which is spreading along with other diseases which are zoonotic in nature, bird flu being other, even though it's a viral infection, there is, because of these filthy conditions, we have approximately 85 billion land animals, and approximately, I think, 80% of all the antibiotics produced goes to livestock. Is there any concerted effort regarding transitioning from that practice? Because I'm wondering if all the 80% of the antibiotics being fed to prophylactically to these animals meant for slaughter, then humans are getting that slow dose of antibiotics, and we are developing uh, antibiotic resistant strains, be it for MRSA or VRE. Are we
Yes,
changing our system to
yes,
a more plant-based system?
yeah,
Go ahead.
yeah. Thanks for the question, and absolutely, that's uh, really an important consideration. And as the caller is mentioning, antibiotics have been used for years in agricultural industry as a growth promoter, based on the observation that when you give it, they get bigger, faster, and so more meat faster. Basically, there not too long ago though, I think both the EU and the United States did agree to dramatically curb this. I'm not an expert on this, but I know those agreements are out there. Maybe they're being phased in, or maybe they're already in. So that you're absolutely right in that it can and was and did drive bacterial resistance, and so that that was recognized pretty quickly as a major source of that. So it's recognized, and I think it's been addressed, but it's probably not down to zero.
I mean, we still have approximately enormous amount. I guess from some statistics, about 70 to 80% of ractopamine, which is being administered to these animals prophylactically, as you validated. I'm wondering what is the organization which should be looking at, because I was reading some stats that antibiotic resistance could become a silent epidemic in the upcoming decades. Is there
Oh yeah.
any governing body which? Go ahead.
Yeah, no, and that's definitely true. I mean, um, so I don't know the government bodies. I mean, I think the USDA I know has been on it, and the equivalent in the EU. And like I said, I know they issued resolutions and proclamations to curb this practice, and I do think that we're a ways into it, in terms of the time post those proclamations. But like I said, I'm not an expert on it. I'd have to look into it to tell you the specifics.
Okay, thank you. Yeah,
Yep.
Physicians Committee for Responsible Medicine is doing a lot of work regarding raising awareness, and I'm not sure if you have watched this documentary. It's called The End of Medicine, and I can put the link in the chat. So thank you.
Great, thanks.
Yeah, speaking of responsible medicine, you also do research in like re-engineering natural products to make them more efficient. So that's really interesting that you went that route. Do you want to talk a little bit about that? How
Sure.
you are, the quinine, did you do that because of malaria and or was it more for other purposes?
Yeah, more for other purposes. I mean, what that that thread of our research, which has been an important component of a few different discoveries, was the when one's people ask me a lot of times, how do you start? Where do you start to find a new cancer drug or antibiotic? And the starting points are indeed challenging. And I would say in the pharmaceutical industry, as I mentioned at the beginning, they have collections of millions of compounds, and they start literally just screening through them, right? Looking for ones that have some sort of hit in some sort of experiment, maybe a cell death experiment, maybe some kind of enzyme inhibition experiment, and a lot of universities have bought large compound collections, several 100,000 or more. The challenge with those collections is that to a chemist, most of the compounds in them tend to be quite simple in their complexity, meaning that they're easy to make. That's why you can get 100,000 of them or millions of them. But the consequence of that is they're structurally simple. So we got into this area that was mentioned, re-engineering natural products, where we can actually take natural products and we can get hundreds of grams of these that we isolate or can just purchase and convert them in a very small number of chemical steps and the natural products, I should say, are complex molecules by all the metrics that chemists care about ring fusion density and stereogenic centers and things like that. And so we can convert those natural products in a series of 5 or fewer chemical steps to compounds that are equally complex but now are quite different from each other. So we've made over 1,000 compounds in this way as a means just to get at a starting point, get at structurally complex molecules that can then go into our starting point experiments.
Oh, that's interesting. So the focus was more on the complexity.
Yeah.
Yeah, that's really interesting. I mean, going through a lot of compounds is kind of a very traditional antibiotic, buyer back in time started doing that. But then the history of that is quite intriguing, right? Like how in Germany, Bayer and so on, they went through thousands of compounds and didn't find really something effective. And then one researcher in England, by accident, probably not. It's probably just English trying to be very subtle. So by accident discovered then the effective compound, which is kind of going back to the question I had before, but that's a longer discussion if just brute force is really, like machine learning. But it's not pure brute force or
Yeah. I think
that makes this probably the interesting way to go.
Yeah, exactly. That's what I was going to say. Every new tool gets layered on the old ones, and the old ones work for a reason, right? Because they're great. But as you have new capabilities, it's just smart to modernize.
Yeah, this is such an interesting discussion, and I could probably go on, but does anyone have like a last question, because the hour is up in 1 minute? So if anyone has a last question for Paul, please go ahead. Yeah, please flash your microphone. Maybe what are you working on in the future? Like related to this project, because I guess you could talk about for hours what you're doing for the future. Like, what's the next step? Are you planning on finding more compounds? Are you checking out for also, like you did for your cancer compounds,
Yeah.
to go into clinical trials?
Yeah, I think all of the above. I mean, we really want to push them forward once we make these discoveries and really push them to benefit people, right? That's what we're there to do. So, and that's a different challenge than the initial discovery and pulling together a lot of different groups and resources. So that's one aspect. But we've also been really taken with this idea of microbiome sparing antibiotics, and I think I'm hopeful that the next wave of antibiotics will be ones that are way easier on the patient, where you don't get these secondary infections. I think that will make a huge difference. And so we have a few other targets that we're excited about, and that also the compounds that hit those will also be gut microbiome sparing. So I think it's really an exciting time for this field.
Oh yeah, that is really interesting. We had a few rooms with researchers about microbiome in humans and also in forests and so on. I think it's an exciting new research area that we are learning a lot about. Which ones are good, which ones are kind of bad and important. It's really exciting. So, well, maybe you come back one day and talk about that.
That'd be great. That'd be great.
Because yeah, it's so much that we still have to learn. Because taking antibiotic is like a huge fire in the forest, basically, probably
Right.
for the gut. So it's very relatable. And then fungi also play a role, which I find play also really interesting roles. Also in cancer now came a paper out how cancer is found in how fungi are found in cancer. So we could talk on and on. So I'll stop here.
Yes, can we please do that? Yeah, I'm interested in recolonization of the gut, for example, after antibiotics.
Yeah.
And if there's any, I don't know if there are lingering parts, how viruses can leave little bits around. And for example, if somebody's had C. diff and then they're treated and then years back, is there any chance that that would return because of prior infection?
Yeah. I mean, I think the effects, the devastating effects of a dysbiosis of the gut microbiome are just, I think we're just starting to recognize. I mean, it's now been linked to colorectal cancer, several other types of cancer, all sorts of, even autism, all sorts of things seem to come back to the gut microbiome. Certainly secondary infections like you're mentioning, and certainly when you get those, then, I mean, some of those, so the recolonization, as I mentioned, now is through a fecal transplant, and which has its own challenges and issues and is not always effective. So I think it would be, obviously, it'd be great if you were never in that situation in the first place, right? If you can make a strategic strike instead of a carpet bomb.
Yes, I thought that strategic strike was really a meaningful and important direction.
Yeah.
And it's also just speaks to one of the million benefits of breastfeeding and breast milk, being a living resource, that it does colonize with so many beneficial bacteria and
Sure.
and I guess, could also be, I don't know, maybe somehow nicer than a fecal transplant somehow.
Well, can you freeze your own gut microbiome? Maybe that's a real company idea.
Oh my gosh, right.
Or
there you go.
if you get or something, or a severe infection, you can just take your own microbiome again, or your mother's or your brother's. I don't know. It'd be funny. So not just cord blood, you get the microbiome freezing. I don't know. Well, so thank you so much for coming. This was such an interesting discussion, and we wish you all the best and all the grants for your work. It's so important. And thank you for doing this work and for getting into this science career. We'll all benefit.
Thank you. Thanks so much for having me. I appreciate this invitation.
Oh, yeah. And it's not altruistic at all to say we wish you all the best.
Thank you. Thank you.
And yeah, enjoy the rest of your day. And thank you everyone for coming, asking questions, participating. If you like discussions like this, follow the club. I think you then get notifications if you opt to get them. And we'll have some more rooms this week about how 1 hour in nature decreases amygdala activity, and Dr. Tarduno, the origin of the inner core structure of Earth, a little bit different. And then, next week we'll have astrobiologists talking about the discovery of origins of life. Very easy questions to solve. So Yeah, thank you for being here, Paul, for participating. We really appreciate it, and I hope we'll hear you back one day. Thank you.
Thank you. Thanks, everybody. Bye-bye.
Thank you.
Okay, I'll close the room in 3, 2, 1. Bye, everyone. Thank you.
Bye-bye.